Hi, I'm Dr. Stacy Blaine from Concarlo, and we are a preclinical oncology company, and we want to transform the drug-resistant metastatic breast cancer space. And we're going to harness the power of biology to combat drug resistance so that we can increase the overall survival of our patients. And so despite advances in treatment, cancer and breast cancer in particular remain unmet needs. About 270,000 women will be diagnosed with breast cancer in the United States this year, and about 40,000 will die. And these are our sisters and our mothers and our daughters and our friends. But the impact of breast cancer extends beyond that, because as women, everyone, we worry about getting breast cancer, and do we have breast cancer? And we want to offer additional solutions to increase the survival for our patients. And so targeting CDK4 and CDK2 at the bottom of this slide have really been the pièce de résistance in the oncology field since the inception of targeted therapy. We think about the drivers of cancer in a funnel, and lots of the drivers at the cell surface and then in the cytoplasm, they all feed down to sort of these ultimate regulators, CDK4 and 6 and CDK2. And while we have many drugs that are effective targeting at the cell surface and targeting in the cytoplasm, if we had good CDK4, 6, and CDK2 drugs, we could render many of the drugs on this slide redundant, and more importantly, we could deal with the back-end resistance that we see in the presence of many of these drugs. And so the CDK4 inhibitors came to pass in 2015. These are great drugs. There's 3 of them: palbociclib, abemaciclib, and ribociclib, very large market space. And they essentially double the remission window for women taking these drugs. So in the metastatic state, if you were going to get 1 year of remission, you might get 2 years on one of these drugs. The problem, however, is that for most patients, the overall survival is really unchanged. And that is because women taking these drugs will eventually become resistant to them. And in order to deal with this resistance, we have to target the drivers of that resistance phenotype. We now know how resistance occurs. So while palbociclib hits the CDK4 target, with time, CDK4's twin, CDK2, is able to rise up and do CDK4's job. Now, CDK4 and CDK2 are not identical twins, but fraternal twins. And while palbociclib continues to work on CDK4, it is ineffective against CDK2. And so in order to deal with this resistance, we have to drug CDK2. The problem, however, is that drugging CDK2 has really confounded our field for several decades. And the reason for that is that it is very similar or identical almost in some cases to other essential kinases. And the drugs that we've developed against CDK2 have met with unacceptable levels of toxicity. So at Concarlo, we took a different approach. We said we're going to target P27, which is the one common denominator in these 2 different types of complexes here in this slide in blue. And P27 is actually an interacting protein with CDK4, 6, and CDK2, and Concarlo's drug IPY is the first P27 specific inhibitor, which will indirectly turn off CDK4, 6, as well as CDK2. How does P27 do this? We think about P27 as a door, and it sort of holds these complexes together, and when it gets phosphorylated or modified, the door swings open and the CDKs are on. But when P27 is not phosphorylated or modified, the door slams shut, turning the kinases off. And Concarlo's drug IPY actually slams that door shut. IPY is a peptide liposomal formulation, so it's based on the naturally occurring P27 inhibitor. And we've just bioengineered that inhibitor to be more stable, increase its PK, and a smaller form that can be run off the peptide synthesizer, so it's a commercially viable product. And what we like about this is that we're harnessing the power of biology. We're using the cell's own way to inhibit uncontrolled proliferation. We packaged it in a liposome. It's a cation charged liposome, similar to the liposomes that many of us had injected into our arm in the last year in the form of the vaccines. And we have confirmed that we can get about 10% of our payload delivered specifically to tumors. We're now in manufacturing with this product with the goal of being first in human in 2023. So At first glance, you might say that using a peptide approach is perhaps not the easiest route to take, but I'm going to argue that there's several serious advantages by using the peptide approach. The first is just the specificity that we achieve. It's really unmatched by small molecule inhibitors, and you can think about biology as sort of a zipper with its target, and so we are getting very specific interaction with P27 that then will inhibit CDK4/6 and CDK2, and that has just not been able to be achieved by any of the CDK2 small molecule inhibitors, and so we think that this will enable us to have less toxicity. The second is this peptide is just not really stable in non-tumor cells, and so this again increases the therapeutic window for this peptide. It is specific and it causes effects in tumors, but really doesn't have an effect at those same concentrations in non-tumor cells. So our intestinal cells and our hematopoietic lineages, they're really not affected by this treatment. And the third, and I'd say really spectacular effect about targeting P27 is that unlike the CDK4 or the CDK2 small molecule inhibitors, which just cause tumor stasis, they slow the tumor from growing, treating tumors with IPY actually kills the tumors. And we now know that in order to really get those increases in overall survival, we have to decrease tumor burden. We have to decrease cause tumor regression. The form of tumor cell death that IPY causes is necrotopus, and that is also a strong stimulator of the immune system. So that leads to the fourth advantage of this form of therapy, which is that we think we can convert traditionally cold breast cancer tumors into hotter tumors, and you have some engagement with the immune system, and perhaps combination with immunomodulator therapies will actually increase our response rates. So it's packaged in a liposome, and so basically this protects the peptide on its way to the tumor. It will circulate in the bloodstream of the patient, and then when it finds a tumor, because of this sort of leaky vasculature or the holes in the blood vessels that is surrounding the tumor, the peptide can sneak into the tumor where it will actually kill that tumor cell, and then that will in turn stimulate the immune system to continue the response. We have intellectual property, both composition of matter as well as mechanism of action on this peptide approach. And we also have a biomarker to help identify patients that would be responsive to this approach, and we have intellectual property on that as well. So because I'm a scientist, I want to show you one piece of data and show you what the power of P27 targeting can achieve. This is a palbociclib-resistant breast cancer mouse model. And so we're plotting the overall survival of the mouse on the x-axis. And when we treat this model with the standard of care for these drug-resistant patients, palbociclib, you can see that all of the animals are dead by day 20 of treatment. But when we treat them with our drug, IPY, or treat them with IPY plus palbociclib combination, you can see that we can extend the overall survival well over 50 days or 100 days in the combination. And I should point out that we stopped treating these animals at around day 35, and their survival continued even in the absence of treatment. Of course, we would continue treating our human patients, and we would expect that we could even extend these overall survival rates significantly. The point of this is that we have been able to extend the overall survival 5-fold over what we see with the current standard of care for these patients. And this is what we need to see in our human patients. This is the power of P27 targeting. So where are we? We are essentially in our manufacturing right now. We've completed in our breast cancer models all of our proof of concept work. We are now preparing the actual GLP drug, and then we'll roll right into safety toxicity studies in 2022 with the goal of having our IND package prepared and start getting that in order for early 2023. We'll also move to a small phase 1 trial in Australia in 2023 to gain some data in humans. We believe this is a serious inflection point for the company, at which point we can come back to the United States and start phase 1 1B, 2A here in the United States. That is also a good time where we can find strategic partners, big pharma, and actually help finish the development of IPY. So who will be our patients? We envision that we will enter into the market as a third-line therapy for patients in the metastatic breast cancer state who have failed off 2 prior lines of CDK4 inhibitor therapy, so have been on 2 courses of palbociclib and are faced now with just going to chemo. We will be an option that will be less toxic than chemo. The options that these patients are presented in this third-line setting carry a lot of toxicity to an already quite sick patient, and so we think that we'll be able to keep the patients on therapy for longer, and that's how we will be able to cause tumor regression and increase overall survival for these patients at this state. So our modeling suggests is that if we capture as little as 15 to 20% of these drug-resistant patients, we will be about a $2 billion annual stream within 2 to 3 years of launch. But we will not stop there, because while we're going to initially go after this metastatic breast cancer state, targeting CDK4 and targeting CDK2 via targeting P27 has potential in many other tumor types, such as many that are listed on this slide. So those that are sort of intrinsically resistant to CDK4 inhibitors, such as ovarian or triple negative, those exist in a state that's already driven by CDK2, and we're going to have an effective CDK2 inhibitor. But also tumor types such as head and neck and non-small cell lung cancer, melanoma, pancreatic. Those are tumors that are basically driven by, we have good therapies for those tumors in the frontline setting, but there are still significant portions of metastatic drug-resistant patients that occur every year, and we will go after those patients as well, increasing our market share and our addressable market. So what will our trial look like? Of course, we will recruit those drug-resistant patients in the HR-positive, HER2-negative setting that have actually been pre-treated with CDK4 inhibitors. We'll also enroll some triple-negative, some ovarian, and some other tumor types into this safety tox study. And then from that, we will roll into a 1B expansion cohort with either mono or combination therapies. And we will use our very good pharmacodynamic markers to show that we have target engagement and that we actually have reached our target, and we'll use that data to move into to help with our additional clinical trials. So let me stop here and tell you a little bit about our team. So I am a molecular cellular biologist trained at Princeton University, Columbia University, and then the Memorial Sloan Kettering Cancer Center. I started my own lab as a faculty member at SUNY Downstate now almost 20 years ago, and I have been thinking about P27, CDK4, and CDK2 for more than 25 years. The team is rounded out by Lisa Marie Casey, Jason Mraz, and Dominique Bredone, and these 3 have experience starting companies, scaling companies, and running companies. Dominique Bredone is a pharma executive, and he has been working with some of pharma's biggest names over the last 30 years, and they have brought that drug development expertise to our team. Our scientific advisory board is really world-class. We have all the needed skills. Our nanomedicine people, Dan Heller and Young Wu, Shanine Singh and Bob DeVita, our medicinal chemists and our modelers, Andy Koff discovered P27, and Gary Schwartz, Neil Rosen, and Jeff Shapiro. They actually are world-class clinicians that run clinical trials at Columbia, Sloan Kettering, and the Dana-Farber. And it's been really important to us to have these oncologists with us from the beginning, because those will be our end users. They will the ones that will help run our clinical trials, but also are the ones that have contact with the patients that we want to help treat. So we've done everything to date on a very lean, mean raise, about $4.5 million, mostly from friends and family, although we have had external validation both from the New York State Seed Fund as well as some impact funding. We have won our first phase 1 SBIR non-dilutive grant, and we met all our milestones and submitted our phase 2 grant. And we actually have a contract from the NIH Nanoparticle Characterization Lab to help with the manufacturing and the CMC and the IND enabling package that we're preparing. And so we are now raising about $15 million. This will help get us to that phase 1 trial in Australia. It will finish all of the GLP manufacturing, do all our ADME and our DMPK work with our lead, as well as run that small clinical trial in Australia. The goal being that with this $15 million, we will have a serious value lift for the company, and we can come back and either raise more money at that stage to run the phase 2 ourselves, or, as I mentioned, partner with Big Pharma to help them finish getting IPY to the clinic. And so I hope that I've convinced you that targeting P27 is unique, that it can have the unique ability to inhibit both the driver of cancer CDK4 as well as the driver of resistant CDK2. We use the power of the peptide and biology to actually have low toxicity and high specificity, and we actually have created a cell death enabling therapy, so we're actually causing tumor regression. We're raising $15 million now to get to first in human in 2 years, and I hope that you are interested in hearing more about Concarlo. Please be in touch at www.concarlo.com, or I'm Dr. Stacy Blaine at concarlo.com. Thank you very much for your attention.